to introduce our speaker. Hello. I think it's my honor to introduce our speaker, Dr. Vinuthan. He graduated in Veterinary College, Bangalore, US, Bangalore, in the year 2000. Sir has completed his master in US, Bangalore, in the Department of Veterinary Science Physiology in the year 2003. Then he went to University of Sydney, Australia, for his PhD in 2010, and he served as research associate at Molecular Structure Development Genetics MRBC. Also, he served in Indian Institute of Science from 2004 to 2006 and in 2011 also. Dr. Vinuthan held an IPS scholarship which is funded by the Australian Department of Education, Science and Training and University of Sydney for the duration of his PhD program. Specifically, he pursued research developing tissue specific as cell culture and he has developed many animal models to demonstrate bioactive roles of milk-derived proteins. Sir has presented his work in various national and international conferences and he has selected for a scholarship to present at an international conference on milk, genomics, and human health in Dawn, California, USA. He has vast experience in teaching and research for 10 years in different universities and institutions of India and abroad. He has published several scientific papers, abstracts in various national and international journals. He won several awards. I will take a few of them. DBT postdoctoral fellowship in the year 2011 in Government of India. And he has pursued a travel grant award in the year 2010 from California, USA. He won Best Research Poster Award in the year 2008 from Fiji Conference, Australia. Now, sir is currently working as assistant professor and the head of the Department of Veterinary Physiology and Biochemistry in Veterinary College, Hassan. I think it's our fortunate to have such an eminent speaker with us today in this occasion. So I would like to invite our host to welcome you, friend. And then before we get start session, there is a small announcement to the participants need to follow. Please turn off your video and audio and don't share your screen in between the session and type your queries and the questions in the chat box. At the end of the session, you will take into the speaker. Again, I would like to welcome all the participants who have attended the session in different corners of the country. With this, I will hand over the session to our first speaker, Dr. Vinuthan N M. Sir, now session is yours, sir. You can proceed. Thank you so much.
Thank you, Mr. Maziana, for your nice introduction. At the outset, my sincere thanks to our Honorable Dean, Dr. Shivakumar MC, and the Student Alumni Association, Veterinary College, Hassan. Right. So, good evening to all. So, coming to today's topic. So today we have a topic on evaluation of semen in domestic animals. Hopefully I'm audible, right? Right, the question is why semen? Why do we collect semen? Or what is the purpose of collection of semen sample? It is not always necessary to check for the semen concentration or to see how the sperms look like. So we might also looking at the other aspects like looking at the infection, then the cancer, and most often in the clinics. So we'll be looking at the sperm count and sperm morphology in order to send it for artificial insemination. So overall, so the semen collection, it is of diagnostic value, and it is also a determination of the cause and the degree of pathological conditions associated with the male genitalia, right? And also it helps to predict the fertility of male, right? So further, so it also helps in detection of how much, at what rate we have to dilute the sample. Okay, the semen, as you know, it's also called a seminal fluid. It's an organic fluid that contains spermatozoa. It is secreted by the gonads and can fertilize the female ova. So if you look at the seminal fluid, it contains several components besides spermatozoa, so like proteolytic enzymes and other enzymes, as well as fructose, so which are very, very important for the survival of spermatozoa. So sample composite, if you look at the ejaculate, the composite, it has three portions. Okay, the first is the pre-sperm, second the sperm-rich portion, the third is the prostatic fraction. So the first, the pre-sperm, it has no sperms or maybe little sperms, and it bit cloudy. Okay, looks cloudy. Whereas the sperm-rich, it is self-explanatory. So it contains rich amount of sperm, and it is somewhat milky and creamy color. Okay, the third portion, the prostatic fraction, it is contain little sperm, or it mainly it contains a volume, so it adds volume to the ejaculate. Okay, the sample volume. So in case of bulls, ram, and bucks, we collect all three portions. That's pre-sperm, sperm-rich, and the prostatic portion for analysis. Whereas in case of dogs, stallion, and boar, we collect prostatic fraction, especially the third portion separately. So why we are collecting this? So separately means in this case in dogs, stallion, boars, we just evaluate only the sperm, pre-sperm, and sperm-rich portions together. That's it. So the answer is, so if you guys, it is this graph is a generalization of semen volume in different species. If you look at that, in case of stallion, boar, and dogs, so you see there will be 8 semen volume, right? So when compared to these other species, bull, ram, and buck. But when coming to this, look at the concentration in millions per ml, so you can see that in this case, in stallion, boar, and dog, it's quite low. That's why. So we don't go for that dilution in this case. So after collection, so we directly inseminate the ejaculate in these species in stallion, boar, and dog. Okay, so requirement for semen collections. So first thing is, so the sexual arousal of the animal is very, very important. So the sexual arousal or excitement is brought about by the sight, sound, smell, touch of the surrounding environment. So, and you can collect the semen by either live mount of the animal, so allowing the male to mount on the female or the teaser animals, or you can go for alternative mounting like using of dummies. So this is commonly practiced in bull, stallion, boar, and mare. Important point is, so when should we be collecting the semen from these animals? So the recommended age of collection in case of bull is 12 months. In case of boar, it's 6 to 8 months. Ram, 6 to 9 months. Stallion, 20 to 24 months. And in the dog, 8 to 12 months. So here, this is quite important. So as in the young animals, so the sperm motility will be comparatively low. So that's why we recommend to collect in the adult animals. Okay, so this is the overall pictures you can see over here. So the concentration wise in different species. All right. So if you look at the concentration in these ruminants, in case of dairy cow, it's the dairy breed, so 2,000 millions, and in average in the beef it's 1,500 millions per ml. In case of sheep, it's around 3,000 millions per ml, the concentration wise. Whereas in case of swine and ours, even though we get high volume of around 150 to 300, we have the less concentration, right? So and for considerations, we need to look at the motile sperm percentage. This is quite important. So if you look in all the species, so the 40% is a minimal thing. So the percentage that should be there in this semen sample, okay? And the normal sperm should be of about 70%. It's the minimal one, okay? 70%. Anything below the 70% normal morphology, so that semen should be discarded. Right. So coming to the different methods of semen collection. So there are various methods, okay? So available depending on the species. Okay, the first method is by the artificial vagina method. The second, the condom or the breeding bag method. This is commonly practiced in equine. And third is the rectal massage method through ampullae and seminal glands. Okay, fourth is an electroejaculation method. So this should be never be done on stallions because these in stallions they cannot handle these electrical stimulations. Okay, right? So, and the fifth method is the recovery of the semen from the vagina. And in case of dog, so we go for like digital manipulation of penis in those dogs which are quite shy. Okay. Coming to just a brief introduction, details about the artificial vagina method. Here it's an ideal method. So this is the common method employed in across all the species. Right, this method is safe for sire as well as even for the collector. Okay, so this is the most preferred method of semen collections. So this is the artificial vagina, where we will be guiding this artificial vagina to the penis when the male mounts. Right, so if you look at this artificial vagina, it consists of a rubber tube. It is lined with a water holding jacket, and it is maintained at around 42 degrees centigrade. Okay, so and at the end there will be a receptacle for collecting the semen, and it is attached at the end of the tube. Okay, this is how the artificial vagina look like. It's a simple instrument. Okay, so before collections, during artificial vagina method, so we need to allow the animal, the bull, to go for false mount. This is quite very important. So at least 2 or 3 false mount is very necessary to excite the bull. Right. So here you can see that. So allow the animals to mount on a teaser bull, and when the penis get erected, directly you can divert the collector. Can divert the penis to the artificial vagina, and you can collect the semen. Okay, that's very important. So these are the images showing the collections on a live mount using the teaser bulls. So we need to be very careful while using the teaser bulls. If it is a female, so see that it is not an estrus. Sometimes what happens if it is not properly guided, sometimes there will be chance of the female is bred. Okay, so you need to be careful. Or sometimes there will be chance of acquirement of diseases, venereal disease from the female. So the collector should be very attentive while collecting from a teaser animal. So, or by the use of dummy. Okay, so these are the images. So where the dummies look like the actual female, and the bulls are excited. So that's how we can collect. So this is commonly practiced in other species like boar and ram. Okay, use of dummies. So in the case of camel, as you know that in the case of camel, a natural mating occurs in the sitting position. Hence, we collect the salmon. At the same position, either using female camel, or we can go for like artificial dummy. Okay, so semen is collected using artificial vagina, so with rubber liners similar to bull. The second method, the condom method or breeding bag method, this is most commonly applicable to equine. Okay, so here what we'll do, we'll place a special rubber bag over the completely erected penis of the stallion. So before mounting, okay, so there will be complete erection will be there. Then it's easy for the collection of the semen. So this method can also be applied to other species. So here what we do is, so we'll insert the breeding bag or the rubber bag into the vagina. So before mating, and we collecting it. Okay, in other species. But in case of equine, so when it mounts, so we'll directly apply the rubber bag over the penis, erected penis. Okay, so coming to the third method, the rectal massaging to the ampulla and seminal glands. So here the manual massage of the axillary genital glands, especially we go for like massaging of the seminal vesicles and ampullae, so which results in the dribbling of the semen. So but here we don't see that erections. Okay, so this method is commonly practiced in the bulls, those which suffer from lack of or loss of sexual desire or impotential coending. Okay, or we can also use this method to collect semen from those bulls which do not respond properly to the artificial vagina method. Okay, so in those, that's the thing. So some of the disadvantage with this method is, so the semen sample collected will be watery and with low sperm cell, right? And there will be a high possibility of contamination by the urine and the diet and smegma, so thing, etc. Right? So this is commonly practiced in bull and okay. So the fourth method, the electroejaculation method. So this method is applied when the males they won't or can't mount, okay, in those animals. So here some of the drawbacks is sometimes the ejaculate may get often contaminated with the urine, and it will be of poor quality. So this electroejaculation method is employed in rams, boar, and beef bulls. So even this is one of the common method. So we normally use in wildlife to collect the semen from the wildlife species and exotic animals. Okay, where we go for anesthetizing the animal. So then we by using this electroejaculation method, we collect the semen. So in this method, the method here, so we're going to insert a bipolar electric probe into the animal's rectum. A large voltage of electric stimulation is given for 2 to 4 seconds at 10 to 20 seconds interval, so until we get the ejaculations. So this method is quite painful, but it should be like done in only in the extreme cases. Okay, so already mentioned this method should not be used in case of stallions. Right. So you can see here this is an electroejaculator where it is inserted in the rectum of the boar, right? And when the current is delivered, so the animals immediately ejaculates. So even in the human pets also you can use, or even in humans, those in man who is like to have child, so there also we use this method, electroejaculation method. Right. So the next method of semen collection is from recovery from vagina. So here what we'll do, so we'll first allow the males to mount. Okay, I have a natural cervix. Okay, and we directly collect the semen from the anterior vagina. So by aspirating into a syringe by means of glass or rubber catheter. Okay, so somewhat this method is also usually is not that good. So it is usually contaminated with the secretions of the female, and there is a greater chance of spreading of diseases from the female. Right. Some operators they prefer this method with sheep, but this method can't be used in case of swine. The sixth method is the digital manipulations of the penis. This is commonly practiced in dog. So what we'll do is we'll slightly apply the pressure and massage the penis, and till the animals get excited, right? So and later we can use AV, artificial vagina, for collections. All right. So here sometimes teaser which may also be helpful. Okay. So it may excite the female. Okay. Excite the male, and so there'll be chance of getting more semen volume. Right. Right. This is in this chart. You can see the overall the methods adapted in different species. So you can see over here case of artificial vagina, it is employed in all the species. It can be employed in all the species. The breeding bag method, so it is particularly in case of stallion. So ejaculation method, it not be carried out in except the stallion. So we can use it for other species. Rectal manipulations, the commonly practice in case of bull and camel bull. Collection from vagina. This is carried out in all the species, like except the dog and boar, right? So then digital manipulations. So it is mainly employed in the dog, and even sometimes, even in case of boar, we can use this digital manipulation, right? So these are some of the techniques of semen collections from different animals, different species. Okay. So now coming to the actual picture, so the evaluation of semen. So before evaluating a semen, so we need to look for the breeding soundness of the animals. So a physical examination has to be done. So both for the internal and external reproductive structures, whether they are healthy or not. So and we need to collect the semen, and evaluation should be done with minimum delay, so that we have to maintain the quality, right? So if it is substandard, definitely those samples should be discarded. So once we evaluate, once we standardize the procedure for evaluations, so we can classify the animals into three categories. The first, satisfactory potential breeder or unsatisfactory potential breeder, or there is the third classification as classification default. So here we can give a chance, there will be re-evaluations. Okay, so for these animals, sometimes maybe on that particular day it may not be well, so maybe we may give one more chance. So that comes under this classification default category. Okay, so coming to the assessment criteria of the semen. So here in the assessment criteria, we'll be looking at the volume of the semen. It's quite important. So there are so many factors it affects the volume. The color of the semen, so they'll be normal, and also sometimes you may get the abnormal colors of the. So maybe due to so many factors, you may get the abnormal color. Then, so you need to check for the motility. So this is very, very important. This is the index of fertility. So the motility evaluation is very, very important. Either you can go for the mass motility. So we need to do both. We can do both mass motility as well as individual motility of the sperms. We look for those aspects, right? So and the important is the morphology. So morphology of the semen. So it indicates the overall the percentage of the healthy semen, and it indicates the quality of the semen. The concentration wise, so you need to have a minimum concentration, all right, to have a high percentage of fertility. Okay, so these are all the assessment criteria we need to consider while evaluating semen. So we'll start with the semen volume. So the volume of the semen is generally influenced by the age, the size of the animal, the body weight, and the breed of the bulls. Definitely. So age. So I told you in an engaged animal, those are younger bulls. They have less fertile motile sperms. And with respect to the concentration wise, we don't see any difference, but you may get in adults, you may get high ejaculation volume, right? The younger, and it is affected by the volume is affected by the younger bulls or the small sized ones. Sometimes it depends on the hereditary and excessive use, or the how frequently you use these bulls for collections. And sometimes when there is an incomplete ejaculations, that also all these factors, it produces less volume. So for that, so what we can do is we can go for the pre-collection sexual stimulations, either through false mating or by proper restrainment. We can increase the volume of semen ejaculation, right? So these are the ones. You can see over here the concentration I already mentioned. So in case of cow bull, buffalo, ram, and buck, they have a low volume but high concentration of spermatozoa. Whereas in the case of stallion, boar, and dog, so we have high volume of semen but less spermatozoa per ml. Okay, that's important. So coming to the second assessment, the semen color. So normally, if you look at the color of the semen, it's quite milky or creamy samples. So creamy color is most common in case of bull, ram, and buck. Buck, as you know, it's a male goat. Whereas milky color will be normally seen in case of buffaloes. Okay, and this the color is influenced by the consistency, so or the concentration of the spermatozoa. So sometimes you may see there will be abnormal colors. If you find abnormal colors, definitely we need to reject those semen. So the abnormal colors you may see or the yellow semen, maybe it is due to the presence of urine or the pus sometimes, or in case of bull, cow bull, you may see the yellowish semen. This may be due to the high content of riboflavin. Okay, so the reddish semen, it is due to the presence of blood. So it indicates that there may be a damage or injury to the lower reproductive tract. Okay, the pink color, pinkish semen, so indicates the degenerative changes. The brownish color, so it indicates the decomposed blood that may be happen. It is maybe it's indicative of old injury to the upper genital tract. The green color, so it's indicate the purulent discharge, purulent degeneration, indicative of the pathological condition of male genitalia. Okay, so sometimes you may get the clear sperm, clear semen, no sperms at all, a condition known as azoospermia. Okay, azoospermia. So we don't see this. So if you get this abnormal color, so abnormal color semen, so we need to discard those semen. Okay, that should not be used for insemination purpose. Right. So coming to the consistency, semen consistency and density scales. So based on the color and the consistency, we can give a scale from 0 to 5. So we can grade it. So if it is cloudy to watery, if the semen is cloudy to watery, so definitely it's have a low sperm concentration and it is considered as bad quality. Okay. Similarly, if you get the thick milky, so it's fair. So and creamy, it's very good. And if it is thick creamy, so on the scale you give it as 5, and it is an excellent quality semen and can be used for insemination. Right. Coming to the third assessment, the motility. We need to check for the sperm motility, right? So this is done by just an visual estimation of the percentage of the motile sperm. So just by visual, so we can estimate how much the motility. Okay, so this is the common laboratory assay we perform when get the semen sample. Okay, so here while assessing, so we evaluate only one single important sperm attribute. So there may be chance of human bias because we are looking at, so 1% will be by visual estimation we are giving the percentage. So there may be a human bias. So to overcome this human bias, we can reduce the human bias can be reduced by evaluating sperm motion using either photographic analysis, so using cameras, okay, or we can now use this by using the computer assisted sperm analysis CASA systems. So this is very expensive, where this method it utilizes sperm and flagellar kinetics. Okay, this CASA is a very expensive method of analysis of motility. Okay. So I told this motility wise, we can either go for the mass motility or individual motility. So mass motility is very, very important. Okay, so it assesses the fertilization capacity. Uh, so we can see that there will be like, once you get the sample, so take the sample on the slide, so and we can observe under low microscope, we can see a collective movement of the sperm cells, so which have the wave motions. Okay, so that's called sperm motility. So this sperm mass motility, it indicates both the concentration and the viability of the sperms, right? So remember, all motile spermatozoa are live, but all immotile spermatozoa are not dead. That means, so even if you look at these spermatozoa at the epididymis, card epididymis, they are immortal. So when the during ejaculations, when they come in contact with the axillary secretion, axillary gland secretions, so they attain the motility. So or by adding diluents, so we can bring back them live. So in case, so if you get the motility, sometimes what happens, you may get low motility. That may be due to if the animal is ejaculated for the first time, or if you are given a long sexual rest for that male, so we will get a poor motility and we may get more dead spermatozoa. So they say that the spring season is best for good quality. So in autumn, so we may see a less quality salmon because of the temperature, okay, variations, right? So evaluation the semen, we can evaluate the gross motility. So it gives a index of sperm activity. So if it is more motile, or with that, definitely it is a good quality one. So in that we may see a frills. So we can see that the sperms moving in a twisting motion, twisting motion, or they should show some progressive movement, the forward movement. So these are the two important criteria we consider while assessing the gross motility of the semen. So this mass motility can be used only for concentrated samples, the need samples. So directly get the semen, collect the semen from the animal bull, and directly you can place a drop of the semen on a warm slide, so with no cow slips, and you can evaluate under low magnifications. Okay, this is the simple technique, right? So we look for these frills or the progressive movement. That's quite important. So we need to remember. So while looking at the mass motility, so look for these frills or the twisting motions and the progressive movement of the sperms. So we can grade it, and this mass activity can be graded by using a rating system. Here we can rate from 0 to 5. So if you want rating is 0, so that means there is no swells. That means no nil or no sporadic oscillation of individual sperms. So 1, so there will be no swells, but we may see the oscillation of individual sperms only. So, rating 2, we can give very slow distinct swells. We find the number rating 3, we can give for slow distinct swill. 4, so moderate fast distinct swill, and we can rate 5, it's a fast distinct swills, and it appears of a good quality. So it indicates it's a good quality semen. So based on the progressive movement, so we can score the semen. So if 0, no sperm movement; 3, 1, 2, 3, 3 for forward progression at a moderate speed. The 5 we can give if we find it's a very rapid progression. So those things. So depending on the progression, we can grade it, right? Scoring from 0 to 5. So after that, we can go for the initial motility of spermatozoa. We look for the individual spermatozoa motility. Here it's in ideal. So here what we'll do, we'll take the percentage of the motile sperms, and it indicates the vigorness of the sperms. So here we use for this to check the individual motility. We'll dilute the semen. It's 1:100, and we observe under high power. Again, we can score it like we can give score from 1 to 5. So if the motility of the sperm, it is forward and progressive in all directions, so we need to check this. So it's highly vigorous. If we are highly vigorous, so we can give it as 80%. Those like the motility of 70 and above 80% will grade as excellent. If the individual movement, individual sperms is showing forward, progressive swelling activity. So if the motility of the overall sperm is around semen is around 70, 60 and above, so we consider as very good. Score 3 if it is forward and progressive, very rapid. So if the motility of the spermatozoa is around 50 and above, 50 to 60%, so it's considered as good. The and again, so if it is above 50% motility, and if it shows the forward and progressive, so we'll consider as a grade. And if the semen are for 50% motility and it is showing some circular or static movement with less progressive movement, so we'll grade as poor. Okay, so this is the one. So see, this is the video. We can. So this is the my. So zoom into higher magnification, you can observe that just concentrate on a single sperm. So we can see that they are moving forward, the progressive movement, and we can see the swells, the twisting movement of the sperm. So it indicates that these are very good quality semen. Right? And you can see over that most of them, they are movement, they're showing the forward progressive movement. So and a lot of swells. So we can consider it as a quality, a good quality semen. So coming to the next assessment, the sperm concentration, right? So we have looked at the volume of the semen, the color, so then the motility, then comes the concentration, sperm concentration. So this is a simple method. The most common means of determining sperm concentration is to just it's simple. Just count sperms under microscope. So with an aid of hemocytometer. So I think you have seen this Nuber slide. So this is this method is commonly employed for counting blood cells. So this works fine for even counting sperms. So the method in brief, just take a semen sample is diluted at 1:200 in diluting medium, and this diluted semen is taken. So in the pipette. So then we need to charge it and charge the Nuber slide. So we and count the number of cells in the 80 square here. So we're counting the sperms in the central primary square. So you know the central primary square. So normally we use for counting the RBC. Similarly, but in this case, while counting the sperms, so we will be counting all the 16 chamber, 16 secondary squares. So which has each secondary square has 25 tertiary squares. Totally, so we will counting the 80 squares, and the sperm concentration per ml is calculated by the formula 10 into number of sperm cells counted into 10 to the power of 6. Okay, so you can see over here the central primary square, and we need to count from all these 16 secondary squares. So alternative method to count the sperms, or the spectrophotometric method, or calorimetry method, or photoelectrical method. So when because the counting sperms using a hemocytometer it's a bit tedious. So there are so many other alternative methods. So in the spectrophometer method, so it measures the amount of light absorbed by the sample, and the more the sperm in the sample, that means more light is absorbed. Okay, so by alternate methods we can count the concentration of the semen. So coming to the next assessment, the sperm morphology. So to check for the normal morphology of the semen, so there are so many different staining techniques, so that may be employed for examining the normal sperm morphology. The common method, the common technique is the Nigrosin eosin stain. Okay, it is quite simple, it's effective, and we can readily visualize the sperms, live and dead sperms. Okay, so that's why this Nigrosin eosin staining technique is also called as live dead stain. Okay, this staining technique, it allows not only to assess the membrane integrity, at the same time we can see the morphology of the sperm. So similarly, other techniques, staining techniques we can adapt, like a rose Bengal stain, Williams stain, or Loftus methylene stain. Okay, but the commonly employed stain is the Nigrosin eosin staining technique. So, so in this technique, eosin and Nigrosin staining technique, so just a simple one. It's just a simple technique. I take a drop of semen, then add 5 to 6 drop of stain, mix it, then make a smear, air dry it, just observe under microscope, right? So if you see the pink cells, pink spermatozoa, the indicative of dead sperms. If it is white, so it's a live sperms. So nearly we can count around 200 spermatozoa, and anything like about 20% dead is not good. So up to 20% is a permissible level. Okay, percentage. So live dead percentage is around 20%. That's it. So that's the simple method to know the live and dead sperms. So coming to the checking the morphological abnormalities. So evaluation of sperm abnormalities, sperm morphology is an important part of breeding soundness examination. So in this case, what we'll do, so we'll look for the size, shape of the head, midpiece, and the tail of the sperms. And also it gives an additional information like integrity of the acrosome and the sperm membranes. Okay, so one, look for the live and dead. So secondly, look for the any abnormalities in the either in the tail region or mid piece or the tail piece, right? The semen sample showing if total abnormalities more than 15%, that should not be used for artificial insemination, right? So permissible up to 15 to 20%. Okay, that's fine. Permissible level of sperm abnormality. If anything more than 20%, so that semen should be rejected. So out of this 15%, I'm telling about the 50% here. Out of this head abnormalities should not exceed 3 to 4%. Mid-piece abnormalities should not exceed 4 to 10%, and the tail abnormalities of about 10% is permissible. That's it. Okay, so generally head abnormalities are considered to be more serious than other sperm abnormalities, because as you know, the sperms contain the genetic material. So any damage, any abnormalities, more abnormalities with the head part, so definitely we should not consider those samples, and the permissible level should be less than 4%. So these are some of the photos showing the abnormal spermatozoa. You can see over here the detached head and the bent tail piece or mid piece. So in this photo, you can see that there's a coil tail sperm. So in this coiled mid piece. And also one of the sperm, you can see over here, there is a presence of a proximal droplet, cytoplasmic droplet present at the mid piece. Those are all And some of the abnormality includes the double head. You can see over here the double head. So the pyriform or the pear-shaped head, and also in this sperm, you can also make out the abnormal midpiece. So there is all the abnormal spermatozoa, and you can see over is the elongated head and the pyriform head. Okay, so we have to grade it. Any abnormalities more than 50% should not be for insemination. So there are some of the tests to assess the acrosomal integrity. So either you can go for staining, or there is a fluorescent. You can see over here. We can go for the fluorescence technique. So where this is the micrograph, fluorescent micrograph of sperm stained with propydium iodide and red patient sativum agglutinin PSA green. So you can see over here the PSA staining with sperm with an we can see over here the intact acrosome. Okay, this is one of the good techniques to assess the integrity. Okay, acrosomal integrity, especially the fluorescent techniques. So the another method to detect any damaged DNA of the head region. So here you can see that in this photo, you can see that the DNA damage can be assessed by acridine orange staining, where here the green spermax shows normal DNA, while there's yellow to red, so they indicates the damage to DNA. Okay, these are some of the laboratory techniques to assess the integrity, acrosomal integrity. So coming to the one more viability test, this is commonly practiced in humans. There is zone of free hamster egg penetration test, or called as EP test. So it's a simple test actually. So we are here in this case we use zone of free hamster egg, so which resembles human ovum. A sperm, in this case, a sperm is capable of penetrating the egg, showing its fertilizing capacity. This test is quite expensive and not reliable, so it's not commonly adopted in a case of veterinary field. Okay, one more test, the OS test, or hypoosmotic swelling test. It is relatively a new assay to evaluate the functional integrity of sperm's plasma membrane. So here, the important principle here is, that the fluid transport across the intake membrane under hypoosmotic conditions causes swelling. Okay, so there will be the equilibrium. There will be swelling. So due to the influx of the fluid, especially the in this case the sperms, so it will expand and bulge, especially at the tail region, and we can readily observe by phase contrast microscope. So it's a simple test indicating that everything the cell is intact. Okay, the hypoosmotic swelling test, OS test. So coming to the last part, the special biochemical test. There's so many biochemical tests, but the important ones I'm going to tell you here. The simple one is the methylene blue reduction test, called as MBRT test. So here, this test is based on the reduction of methylene blue. So it depends on the dehydrogenase activity of the spermatozoa. Here, hydrogen ion is liberated under anaerobic condition for a unit of time. It's a simple test where we can, if there is a higher concentration of the active spermatozoa, so that will cause that will reduce the methylene blue in less time. Okay, so we can grade it. So if it is a good quality sample, so the methylene blue reduces within 3 to 5 minutes. Average samples it may require 9 minutes, and if it is a poor quality, it may take more than 9 minutes. That means, so it indicates the concentration of the active spermatozoa. Okay, so one. And finally, the fructolytic index. As you know, the fructose is a reduced sugar that present in the seminal plasma. So here in this test, so we're looking at the rate of utilization of fructose and formation of lactic acid. Okay. Here it is measured as amount of fructose in milligram utilized by the spermatozoa in hour at 37 degrees centigrade. Okay, these are some of the tests we can employ either at the collection center or even at the field level. Some of these tests you can that is it. Thank you.
Hello. Thank you so much, sir, for your wonderful presentation. I think participants have enjoyed a lot and they have enlightened by this presentation, definitely. Without taking much time, we move on to question and answer session. Sir, can you present Hello?
Not audible.
Hello, sir. Can you hear me? Vinuthan, sir.
Hello.
Hello, sir.
It's not audible.
Hello. Hello, sir.
It is not audible.
Hello. Now it's audible, sir. Hello.
Sir, please increase the volume, sir.
Yeah. Now I can hear. Okay. Hello.
Hello, sir. Hello. Hello. Hello.
Can you hear me?
Hello, sir. Could you hear me? Vinuthan, sir. Abdul, sir. Abdul, sir. Hello.
Hello.
Sir.
Okay, I can hear you.
Now it's audible? Hello.
Okay, Vinuthan sir, can you hear me?
Yeah. Now can I?
Okay, sir. It was a very wonderful presentation. We'll quickly go to question answer session, sir.
Right. Yeah.
Yes. There is a question from Nidhi. Why recovery of semen cannot be done in swine?
Why? The recovery of?
Why
Oh, okay.
semen cannot be done in swine?
It's fine. Okay. So because this recovery semen from the vagina we normally adopt in the large animals. Okay. So, but not in the small like a dog and a boar.
Okay, sir. Priyanka is asking, what are teaser bulls?
Okay, these are bulls. They are not nothing but any bulls, or you can use the females. So, which is the animals like which is used for selection. Okay, so we'll bring the bull to those females or any other male. So it actually teases which helps in selection of the family. Okay.
Okay, sir. Sir, Dr. Srikanth is asking, what may be the reason of ejaculate of bull without spermatozoa?
What percentage?
Two accounts with. Okay, sir. What may be the reason of ejaculate of bull without spermatozoa? This is the question.
Okay, yes. Sometimes what happens when the animal is not aroused, sexually aroused, right? Chance. So we have some animals which are shy, so they cannot ejaculate, right? So that's why I told you the environment should be there to create the excitement in the sexual excitement in the male. If there is no sexual excitement, definitely we don't get the sperm ejaculations. Sometimes what happens, so maybe some pathological conditions of the sperm. It's a condition called, I told you, the potential coindy, it's a condition, so where we don't see any sperm at all in the semen.
Okay, sir. One more question from this way. What is the difference between the primary and secondary spermatozoa defect and or abnormalities?
Okay, the primary and secondary, so we can differentiate. Primary is when it the cause from the any problem with the genital organs. Okay, so the while spermatogenesis, when it's problem with the spermatogenesis, or while of the spermatozoa, the comes gets damaged, so that's called primary abnormalities. Secondary abnormalities, so I think so after collections, whatever the abnormalities they get, that is called secondary abnormalities.
Okay. Okay, sir. Next question from Satish Chandra. How can you separate egg from?
So that is using cell sorter. There is a, I think so that there is by looking at the specific protein. Okay, in the thing. So in this case, sometimes only for sex. That is allowed actually. Cell separation of X and Y. So that can be detected by detecting the HY antigen. Okay, which is present in the male.
Okay, sir. Sanju Kumar is asking, tell me some information about semen straws.
Semen straws. Okay, there are two types of semen straws normally available. So 0.25 ml and 0.5 ml. Okay, and it comes in different colors. So different color indicates the semen from different species. Right? So normally, so what we normally use is 0.5 ml and 0.25 ml. That's two types of straws currently available.
Okay, sir. Dr. Srikanth again asking, are there any toxins or fluids that may cause abnormalities in spermatozoa?
Definitely. Okay, so some degenerative changes. So definitely there is some toxins that may affect the spermatozoa. It's a condition.
Okay, sir. Sir, Sona is asking, in field, which is the most suitable method for semen ejaculation?
So collection. Okay, semen collection. That's every method.
Semen collection and evaluation in field condition.
The collection, the best method is the artificial vaginal method.
Okay, sir. I think her question is most suitable method for semen evaluation.
Okay. So the simple. I told you, you can go for like simple. So look for the mass motility. So it indicates the consistency and the concentration of the spermatozoa. So look for the individual motility, and also the simple test by immunocytometric method, so we can assess the concentration of the spermatozoa. And by all this morphological method also, by looking at the live and dead percentage, that overall gives the quality of the semen. If more than a live percentage, and we definitely the fertility rate should be high. Okay, so all this we can carry out at the field level. So at the VD, we can if you have microscope, the simple thing, just we can even you can assess the semen straws. Take out the semen straws or monthly interval, just one semen straw from the stock. Look for the motility, mass motility, individual motility, the count is concentration, and also look for the lavender percentage. And simple test, I told you simple test for or nuclear reduction test that can also be employed. All the simple tests, so definitely we can improve the fertile percentage. Okay.
Okay, sir. Okay, sir. That's it, sir. Questions.
Okay.
So last question is there, sir, from Nidhisri. She is asking, can we identify that a particular defect in sperm is after collection or it is during spermatogenesis?
Sorry, one more time. Repeat the question, please.
Can we identify that a particular defect in sperm is after semen ejaculation or it is during spermatogenesis? I don't know, sir. There is some confusion in question only.
Okay.
Nidhisri, can ask the question to the sir directly. Nidhisri, you there?
Hello.
Yes, please, Nidhisri, you can ask.
So my question is that while we see the defects in the sperm, can we tell that it is
Hello.
that defect had occurred during the spermatogenesis or it is after the semen collection?
Sir, did you got the question?
No, sir.
Okay, sir. She is asking
Yes.
whether the defect in the sperm can be evaluated after semen collection or during spermatogenesis we can evaluate.
Okay. So I told you the what you call the breeding conditioning. Like the breeding bull we can make out. So whether they are able to erect, erection is there, normal erection is there, or whether they are cooperative. So depending on the health status, we can somewhat assess the we call it as a breeding soundness. Breeding soundness can be assessed by looking at its internal and external structures, right? So if there is any defects in the normal structures, definitely we can make out that there may be chance of abnormalities in the spermatozoa. So hope you got the answer.
Dr. Srikanth can unmute your mic and you can ask the question directly. If the question is different, you can speak it to the sir directly. Please. Hello. Hello.
Yes.
Hello, sir.
Hello.
Sir, third semen and what time it should be used, sir?
Which one? Sorry.
After thawing, sir.
Yes.
Semen, what time it should be used, sir?
Okay, within 40 seconds, 30 to 40 seconds, or okay, after once you get the semen, when you take out the semen from the liquidated, it should be used within 15 minutes. So within 15 minutes, you need to inseminate 15 minutes.
Okay. Thank you.
So thawing can be done at like 30 to 45 minutes. So after that, so immediately, so within 15 minutes, we need to inseminate the animal.
Okay, Dr. Srikanth, please unmute and ask.
Hello, can you hear me, sir?
Hello. Yes, please. Please ask the question.
Sir, she is asking that while we are doing evaluation, semen evaluation, can we differentiate that whether the sperms got abnormalities of semen sperms are during the spermatogenesis or during the ejaculation while we are collecting the semen? What may be the reason? Can we identify it? She is asking, sir. Hello.
Okay, Vinuthan sir, did you got the question?
No. I didn't hear it.
She is asking.
Yes, please.
Microscopic evaluation of sperms. Hello, can you hear me, sir?
Hello?
Can you hear me, sir?
Not hearing anything. Dr. Srikanth?
Hello.
Okay, sir, I got the question. What they are asking is whether during the semen evaluation, can we differentiate that is abnormality of the semen is due to procedure of collection of the semen or due to spermatogenesis. Can we differentiate that while we evaluate the semen?
Okay, that's difficult to say. Uh, maybe by looking at these abnormalities, like some abnormalities, like we can't assess clearly, but at some percentage we can make out. So, so if it is the defect of the uh, animal itself, so maybe we may get different abnormalities like what is that, the pear shape or the coil shape, those things. So after collections, so the once you get collections, maybe you may see some while processing, you may get the abnormalities like a detached tail, right? So detached egg, those part. Okay, so by looking at that percentage, we may assess either it's a problem with the spermatogenesis or after the collection.
I hope answer is up to their best level, sir.
Okay, yeah.
Right? Okay, sir. It was a great and wonderful presentation, sir, and we are grateful that you are with us today for nearly 1.5 hours. It was a very wonderful presentation, and there is a well discussion with participant and you, sir. I thank wholeheartedly from Veterinary College Hassan and Student Alumni Association, sir.
Thank you. Thank you.
Okay, sir. I also thank Dean Veterinary College Hassan, M.C. Sukumar sir, who was there with us. He is not there now, but he was there with us before. I thank him for his support and motivating us for beautiful and conveying veterinary education through webinars. I thank you, sir. I also thank different doctors, faculty members, students, and other audience who are present today. I thank each and everyone. Thank you.
Thank you.
Okay, sir. We'll end this meeting, sir. Thank you very much, sir.
Thank you.